Role of calcium in cross-bridge formation. We know that we have head of myosin, and during relaxation, this head of myosin are separated from the binding site on actin or the binding sites of actin. So that is the actin protein, and that is the binding sites of actin. And we know that we have a protein preventing the contact between head of myosin and actin, and this is the thromboyosin protein. Thromboyosin protein. This thromboyosin prevents what? Prevents the contact between head of myosin to the actin binding site. And we have another protein in the thin filament, another protein in the thin filament, which is what? Which is the troponin. Troponin, and this troponin has a specific subtype which is known as the troponin C. Troponin C. And with the release of calcium from the sarcoplasmic reticulum, what's going? Release of calcium, calcium bind with troponin C, with the movement and with the binding of calcium to troponin C, we have conformational changes. We have conformational changes. What's the meaning of conformational changes? The tropomyosin will be moved away, from the binding sites. The tropomyosin will be moved away from the binding sites, and this will allow the head of myosin to come and bind with actin. So again, calcium release from the sarcoplasmic reticulum, bind with troponin C, movement of the tropomyosin away from the binding sites by conformational changes. This allows the head of myosin to bind with actin.